So our next speaker is Frederique Reverchon. She got her bachelor's degree from the National Engineering School of Agronomy in France. Then she went to Wageningen to do a master's degree in environmental sciences. And then interestingly, she did a PhD on studying ectomycorrhizal fungal communities associated with pines in the Institute of Geology of the National University. I didn't know that the Institute of Geology did biological research, so it's very interesting. And she'll be presenting a talk on microbes and microbiomes for sustainable food production. I hope I pronounced your name correctly.
No, but that's okay. Gracias. Good morning. Thank you very much for the invitation. So today I'm going to talk to you about the potential of microbes and microbiomes for sustainable food production. First, I'd like to talk about what we already know about plant microbiomes. The title of my slide is a bit hidden, so sorry about that. Actually, we know quite a lot about plant microbiomes. We know that plants are colonized or inhabited by a lot of different microorganisms, mainly bacteria, mainly fungi, and that the composition of these microbial communities vary according to the plant tissue that we are talking about. They vary in composition, but also in diversity. This microbial diversity being larger in the soil or the rhizosphere than in the inner compartments of the plants or in the phytosphere, for instance. So the structure of these different plant-associated microbiomes differ depending on a lot of different variables. For instance, the soil type, the host, genotype, the plant species, obviously, climatic conditions, management practices, if we're talking about crop-associated microbiomes. So a lot of different conditions influencing these plant microbiomes. There is, however, a core microbiome associated with different plant species, which is good news. We're able to find a set of common microbes associated with the same plant species independently from the different environmental conditions where the plants are growing. And generally, this core microbiome has beneficial effects for the plant. They can promote the plant health, the plant growth, and ultimately the plant fitness. So how are the different benefits that the microbes can play for the plant? First, growth promotions through a different array of mechanisms. We are talking about microbes that are forming symbiosis with plants, nitrogen-fixing bacteria, for instance, or other microbes that have been previously mentioned in the past talk, like microbes able to solubilize phosphorus or produce siderophores that can capture or unlock different nutrients that otherwise wouldn't be available for the plant. So through an enhanced acquisition of soil nutrients, microorganisms can promote plant growth. They can also produce precursors of plant phytohormones and thus promote plant growth. They can help stress control. We've been talking in the previous talk about abiotic conditions that are very stressful for the plants, increasing temperature, salinity, drought, obviously. So microbes are also a sustainable way to be able to alleviate these abiotic stresses, mainly through also the production of plant phytohormones. And microbes are able to defend the plants against the attack of different pathogens or pests. They can do that directly by enhancing the plant defense response, triggering the plant's defense response, also indirectly via their antagonism against plant pathogens. For instance, by secreting enzymes, lytic enzymes, or antimicrobial compounds. And I'm going to talk to you a little bit more about this in the next slides. So altogether, microbes or plant-associated microbiomes have a huge potential to foster plant health. The diversity of plant-associated communities is very important, because by having a diverse plant-associated microbiome, then we have kind of a balance, an equilibrium that is helping or preventing a possible dysbiosis. So there are quite a lot of studies recently, if we're talking about plant health, that are showing or studying the microbiome associated with healthy plants versus diseased plants or sick plants. And what they usually found, let's see if I can find the pointer, yeah, is that the structure of plant-associated microbiomes are shifting when the plant is diseased or affected by a pathogen. These graphs that I'm presenting to you are from a study about beetroot, and they are studying the composition of the microbiome of beetroot when it's affected by different fungi that are triggering beet rot. So here you can see the microbiome associated with healthy plants, and here the microbiomes associated with sick plants in the case of beetroot, which is quite interesting. There are a lot of studies studying this in different pathosystems, different crops, different pathogenic agents, and they all kind of find the same results. There is clearly a different structure of the plant-associated microbiomes when, when the plant is sick. What we know is that not only the structure of the microbiome is affected, but also the composition of this microbiome. So here it's the same example from the beetroot microbiome, bacterial microbiome in this case. And they're showing to us that when the plant is affected by root rot, the composition is quite different of the microbiome than the healthy plants. In this particular case, they associated their microbiome studies with the isolation of different microorganisms, aiming to find some bacterial antagonists from these pathogenic fungi. And so that was interesting. But what was interesting as well is that they wanted to study the microbiome of plants, healthy plants, with a high proportion of antagonists and with a low proportion of antagonists, which are these different points here, the color points, and these two bars here. And they didn't really find a difference in the, I'm going to remove this, sorry, in the microbiome of healthy plants with high or low proportion of antagonists. So why am I talking about this today for sustainable food production? Is that in this particular study, they recommend that we could use microbiome studies to actually propose some microbial indicators of the health status of different plants. So in this study, they sampled a lot of different beetroot fields in Europe, but in different countries. And what they found that there is a consistent response of the beetroot microbiome to this particular disease. And they could find some species, either fungal or bacterial, associated consistently with the disease status, and other bacterial or fungal taxa that were consistently associated with the healthy plant status, which was quite interesting. So we could also use our knowledge about plant-associated microbiomes to try design synthetic communities that are a bit more efficient. Traditionally, we've been using single-strain inoculants for biocontrol or biofertilization, and single strains are okay, but that would be better to kind of try to preserve the initial diversity that the plant microbiome has, right? So the idea is to try to design more efficient microbial synthetic communities that would allow us to reduce a little bit the huge diversity that this plant microbiome have, but preserving some of the important interactions that we can find within this microbial community, and combine different beneficial functions that one single strain maybe could not provide. So there are quite a lot of literature also on this aspect, and how we could use synthetic community syncoms to try to design more effective biocontrol agents, suppressive soils, even try to manipulate the soil microbiome to foster this inherent capacity of the soil to not be conducive to certain diseases, and as biofertilizer, to try to design more efficient biofertilizers. So we could do that, not getting rid of all the isolating techniques that we've been using in microbiology for quite a long time. They're really important. We need strains to work with, but combining these isolation techniques with other techniques, studying genomes of individual strains, studying the diversity, the functional, taxonomic and functional diversity of microbial communities to try to design more effective syncoms. And an effective syncoms, what would that be? That would be a synthetic community that is able to colonize efficiently the plants, and that would persist through all the development stages of the plant, not only for the seed or at the beginning of the seedling stage, but even at the more productive stage of our different crops. And combining, obviously, different functional benefits that they could provide to the plant. So here, this is the end of my introduction. And before I'm talking to you about some of the results that we've been generating in our labs, I want to acknowledge well, some of the students and colleagues that have been participating in the results that I'm going to present. So which crop are we working on? In our lab, we're working on avocado. I'm not going to talk to you about the importance of avocado for Mexico. I think everyone is very aware of it. As it was previously mentioned, the avocado genome has been sequenced here. So I'm not going to talk to you about that. But we're working on avocado, and particularly with two diseases that are affecting avocado. The first one is Fusarium Dieback, which is actually not only a pathogen, but it's a pest pathogen complex, which ecologically speaking is very interesting. So this Fusarium Dieback is caused by fungi of the genus Fusarium, as you could have guessed. And these fungi are vectored by ambrosia beetles. They are borers that are creating garries in the trunks and the branches of a lot of different plant species. Avocado is one of them, but a lot of forest species as well. Other crops such as citrus, macadamia, and yeah, this has been a huge problem in different countries such as Israel, South Africa, the United States, and the beetle has been detected in Mexico a few years back. So it's actually a quarantine. I don't know if I'm pronouncing this correctly. A quarantine disease. The other disease we're working on is Phytophthora root rot, which is very common in almost all avocado orchards. And Phytophthora is an oomycete and is causing rot and a regressive dieback of the tree. So we're working with these two pathogens. What are we doing? Well, first, we studied, as I mentioned in my example of the beetroot before, the microbiomes of plants. Avocado trees were healthy, and avocado trees were affected by these two diseases. First, I'm going to talk to you about Fusarium Dieback. We went to California actually to sample because the disease was not in Mexico. So we went to California to sample in an orchard where avocado trees were either healthy or without any symptoms of Fusarium Dieback and well, affected avocado trees. What did we find? We found that the richness and diversity of the plant-associated microbiome, in this case, the Rhizosphere microbiome of avocado, decreased when the plant was affected by the disease. And not only the diversity, the microbial diversity decreased, but the structure of the microbiome was also affected, as you can see here. No? In red, the red dots are corresponding to the asymptomatic plant, and the blue dots are the symptomatic plants, or Fusarium Dieback affected. No? And we found that either in terms of incidence of or presence or absence of microbial taxa, or when we consider the relative abundance of different bacterial taxa, there was a shift in the structure of the Rhizosphere microbiome in avocado when avocado trees were infected by this disease. What was interesting is that when we studied the same, or we carried out the same kind of studies with Phytophthora root rot, also in avocado, we found very different results, obviously, right? Like this is a different pathogens and its way of action is quite different. But in this case, we didn't really find any impact of the disease on the microbial diversity at the Rhizosphere level, on the microbial richness, and neither on the structure of the microbiome. So here you can see a separation of asymptomatic and symptomatic trees, but this was not significant, so we cannot say that there was a shift in the structure of the microbiome. Then we went to the composition of the microbial communities present which were present at the rhizosphere level of avocado trees. And in the case of Fusarium Dieback, actually, when we studied the microbial composition at the phylum level, we did not find really a clear trend between, well, a difference between healthy and symptomatic trees. There was, however, so we found a difference in the structure, and there were, however, some bacterial taxa which were exclusive of healthy trees and other which were exclusive of diseased trees. So we thought, okay, maybe it's not at the phylum level, but we have to go at lower taxonomical level to find these differences. So we studied the OTU, the most abundant OTU that we found at the rhizosphere level, and we thought, okay, in the most abundant ones, we're going to find these differences that our beta diversity indicated, and we did not find it. These are the 50 more abundant OTUs that we found on the rhizosphere of our avocado trees, and except from one taxa, which was a gamma proteobacteria, I think, there was not any difference in the relative abundance. So this was quite interesting because actually what this study showed us is that the differences in the microbiome when the plant was under stress and affected by Fusarium Dieback were given by rare taxa. And rare taxa is something that we did not really take into account for a very long time, thinking, okay, the abundant microbial taxa are the ones that are interesting and doing all the works. But actually there is a big trend in ecology right now about the importance of the rare biosphere and the importance of rare microbial taxa in particular for nutrient cycling, the health of their host. And here we're talking about avocado, but it can be, it is shown as well for the human microbiome. So we need to take into account these rare bacterial taxa that sometimes we kind of remove from our bioinformatic analysis as well. So that was quite interesting. And then we studied the composition of our rhizosphere microbiome in avocado affected by Phytophthora root rot, and the results were quite contrasting. Since the phylum level, we found differences in bacteria and in fungi associated with the rhizosphere of our avocado trees. I'm going to talk to you a little bit more about that. And also, we found quite a large proportion of OTUs in this case associated exclusively either with the sick status or the healthy status of the microbiome, of the tree, sorry. So trying to think about microbial indicators that could show us, like give us an indication or could act as a proxy to tell us the disease status of the plant. In the case of Fusarium Dieback, we found these taxa, I'm not going to pronounce it because I don't know how to pronounce them in English, that were indicative of the healthy status of avocado trees, and these taxa that were an indicators of the sick status of the avocado trees. And in the case of Phytophthora root rot, these phyla were more represented in the healthy status, and these phyla were more represented in the sick status. So it's quite interesting to see that, for instance, this phyla, Armatinomides, I'm not quite sure, is an indication of a healthy avocado tree when we're talking about Fusarium Dieback. But when we're talking about Phytophthora root rot, for instance, it's an indicator of the disease. No? So this shows us that really this kind of study has to be like the pathobiome or the pathosystem specific. No? We cannot generalize because we're talking about the same host that specific microbial taxa is going to be an indicator of a disruption of the microbial community because it really depends on the pathogen and the mode of action of this pathogen. So we also complemented our microbiome study with the isolation of different microorganisms, mainly bacteria, to try to find some antagonists to these different pathogens. So we isolated a lot of bacteria from our avocado trees in California. We have some from Mexico as well, obviously. And we confronted them against different pathogens, Phytophthora cinnamomi, obviously, Fusarium uwalesi, which is one of the causal agents here of Fusarium Dieback. Fusarium Dieback is mainly caused by several Fusarium species, but also by different Graphium species. So we also tried our bacterial antagonists against these different fungi. This was done by dual cultures, but we are also working with microbial volatiles. I'm sure you've all known about you all know about the potential of microbial volatiles for antagonism of pathogens as well. So we have a collection of different bacterial isolates that are really interesting in terms of the emitted volatiles, because these volatiles are able to antagonize different pathogens as well. Most of these bacteria that we isolated belong to the genus Bacillus or Pseudomonas, which are the easiest one to isolate, kind of, right? We also worked with scanning electron microscopy to corroborate the damage that these bacterial antagonists can provoke in the hyphae of our different phytopathogens. So here are some examples. We see here in the dual cultures, for instance, clear morphological alterations of the hyphae of Fusarium curotium in this case, and vacuolization of this hyphae, the formation of kind of big vacuoles here, or conglobations. In the case of volatiles, the damage that we can observe is kind of different. We can observe distortions, morphological distortions of the hyphae, and a shriveling of the hyphae, which is what you can observe here in comparison to the control, where the surface of the hyphae is more smooth, right? Okay. One interesting thing is that we're also working with chemists based at INECOL to try to understand what are the microbial compounds, the composition, the chemical composition of the compounds that are responsible for the antagonism that we've been observing. So we're working with different bacterial extracts in different kind of vehicles to isolate polar and non-polar compounds. And then we're trying or verifying, corroborating the antifungal activity of these microbial extracts. And we found the same kind of damage induced in the hyphae of the different pathogenic fungi, caused not by the bacteria directly, but at the compounds that are emitted by these bacteria. And what we found mainly in our bacteria from the genus Bacillus is that most of these compounds that are responsible for the antifungal activity that we're observing are cyclic lipopeptides. This is kind of well known for Bacillus species. Cyclic lipopeptides are compounds that are really interesting because they have an antifungal activity and anti-oomycete activity as well against a whole range of fungal pathogens. And we found that these lipopeptides belong to different families, iterine, fungicin, and surfactin. And when the bacteria is able to produce the three types of cyclic lipopeptides, their antifungal activity is actually quite stronger. So these are examples of other extracts. And on top of cyclic lipopeptides, we have found also polyketides and non-ribosomal peptides that are responsible for the antifungal activity that we've been observing before. We've done this study of the chemical characterization of the antimicrobial compounds for the volatiles as well. And we found a different set of volatile compounds that are really active against Fusarium species. Here are some of them. I'm showing here some of the assays that we've been performing with our microbial volatiles. Some of these compounds, we were able to find them in the form of commercial compounds, commercial standards that you can acquire from the different providers, test them to verify that this is actually one of the compounds responsible for the antifungal activity. And one of the most promising compounds that we found is dimethyl disulfide, which is one of the compounds that we have tested with the strongest antifungal activity. So this is what we've been studying for the antifungal activity and trying to find potential biocontrol agents for these two diseases that I was mentioning. Now we're working to try to combine different strains to see if we can maximize their benefits and to assess also their plant growth activity and see if on top of being efficient biocontrol agents, they could act as biofertilizers as well, which could be very interesting. So we've been isolating on top of Rhizosphere microorganisms, endosphere microorganisms, because we found that niche complementarity sometimes is very interesting or very a potential tool to maximize or combine different interesting functions. So we've been combining endosphere microorganisms with Rhizosphere microorganisms and testing them in Arabidopsis. So here we first evaluated the plant growth activity of our single isolates on Arabidopsis thaliana. What we found is that most of our isolates were able to enhance the plant biomass, modify the root architecture, promote the number of lateral roots, which are the absorptive roots, so the ones that are actually interesting for plant growth promotion. We also found that our isolates were able to increase the density of root hair and also the length of the root hair, which is what I show here. We have two endophytic isolates, which are particularly interesting, Pseudomonas. And this growth promotion generally seems to be mediated by an auxin root. So we've been combining these two kind of bacteria, Rhizosphere with Endosphere, showing promising results in Arabidopsis thaliana growing in soil. And we've been working with tomato as well, evaluating different criteria. I'm going to pass this very quickly because I speak slower in English somehow, sorry. So we've seen that our strains, either when inoculated as single strains or in consortia, or mixing two strains together, were able to promote the height of the plants, the biomass of the plants as well. And what was most interesting is they were able to promote the induction of flowering. And our plants, which were inoculated with bacteria in tomato, our tomato plants, were able to produce flowers a month before the control, which was not inoculated by the plant, which was really interesting. And here are some of the results that I'm going to show you from our tomato plant. And finally, as a mean of a conclusion, these results show really the potential of microbial inoculants to try to find or to try to reach a more sustainable food production and more environmentally friendly agricultural practices. So yeah, let's not discard microbiomes in this assessment of soil biodiversity. And with this, I finish. Thank you very much.
Thank you, Fred, for very nice results. I was wondering if you have any data of what happens to the microbiome if you have an avocado plant that is infected by both Fusarium and Phytophthora?
No, we don't actually. One of the problems with Dieback is kind of complicated to study because it's not in Mexico and can work with the same, but it's a quarantine lab at, well, Senacica, and then we cannot perform greenhouse experiments with this. But that would be, yeah, a very interesting study to combine both pathogens and see how the microbiome resists. Yeah, thanks for the suggestion.
I have a question. How likely is that these inoculants persist in the soil? Because there is always a strong competition from indigenous microbes.
Yeah, that's something that we're investigating at the moment. That's a very good question. So something that we did not assess in our tomato plant experiments, for instance, is the colonization capacity of the plants. Some of the bacteria we assessed were working very well together, like in teamwork, but some of them seem to be working better when they were inoculated as a single strain, which was kind of disappointing because we were always thinking about teamwork as the best way to go. So what we wanted to assess is actually the persistence of these bacteria in the soil. And the experiment we're carrying out at the moment, we had a big avocado experiment where we actually infected ourselves Phytophthora cinnamomi. The results I presented you here about the microbiomes, we went to the field and chose our trees, but we couldn't really control many factors, such as, for instance, if the trees were infected at the same time or the amount of inoculum of the pathogen. So we implemented a huge experiment in the greenhouse where we actually inoculated Phytophthora cinnamomi. We knew exactly the concentration of the inoculum, and we monitored the changes in the microbiome across the disease development. And we introduced an extra treatment, exactly what you're mentioning, Luis, about a biological control agent. One of our most promising bacteria that I showed you here, we inoculated it, and we're studying at the moment. We don't have the sequences yet, so I'm not able to actually answer the question. But if you invite me again, maybe next year, then I might be able to show you and answer more precisely.
Thank you very much.
Thank you.
Thank you for the presentation. We go to the last